Good afternoon, children. We are continuing the chapter Cell Cycle and Cell Division from class 11 Biology. In this part, we are going to revise some of the important questions that are important for the coming examination. We have already sent the detailed video of this chapter, Cell Cycle and Cell Division, early in advance. Today, we will be discussing some of the important aspects of this chapter. The first and most important thing is life cycle. What is the meaning of cell cycle of a cell? Cell cycle indicate the sequential events by which a cell duplicates its genome. A sequential events by which the cell duplicates its genome and synthesize other components of the cell, ultimately divides into two daughter cells. Ultimately divides into two or more daughter cells is called cell cycle. So what is cell cycle? Cell cycle is sequential changes in the cell during which the genome of the cell duplicates and the component of the cells are synthesized and ultimately the cells divides into daughter cells is called cell cycle. There are mainly two stages in the cell cycle. They are interphase and mitotic phase. Interphase and mitotic phase. What is interphase? Interphase is the stage between two mitotic divisions. Stage between two mitotic divisions are called the interphase. Okay? During interphase, we can see there are three stages. What are the three stages of interphase? G1 phase, S phase and G2 phase. G1 phase, S phase and G2 phase are the three stages, three steps in the interphase. In G1 phase, you can see the cell continues to grow. Because before this G1 phase, the cell was just, daughter cells were just formed by mitosis or meiosis. And in G1 phase, the cell continues to grow. In S phase, that is called a synthetic phase, the DNA duplicates. The DNA, genetic material duplicate or replicate into two copies. Now G2 phase is the continuation of the G1 phase in which proteins are synthesized. Proteins are synthesized by the cell. You got it? Yes. Now, after this interphase is over, in these three stages, G1 phase, S phase and G2 phase, we can understand that the next one is M phase, that is called mitotic phase. Mitotic phase. Mitotic phase includes we can say karyokinesis and cytokinesis. Mitotic phase includes karyokinesis and cytokinesis. What is karyokinesis? The division of the chromosome. The division of the chromosome or the DNA. DNA copying takes place. DNA duplicates, DNA replicates. So this replication is otherwise called, karyokinesis. Okay? In cytokinesis, the two daughter cells are formed receiving or with one each chromosome that is already formed. Okay? So what are the two stages in the mitotic phase? Karyokinesis and cytokinesis. Karyokinesis and cytokinesis. The details of which we can study from the previous video. So karyokinesis and cytokinesis are the two stages of the mitotic divisions. You got it? Clear. Right. Now the second question. What is Poisson stage of a cell? Some of the cells in the body, for example in heart, after G1 phase they stop the further steps. After G1 phase. They do not divide. They do not divide after G1 phase. And such cells which do not divide and they stop growth and they are in the inactive stage. Such changes in some cells after G1 phase which do not divide and they undergo inactive stage is called Poisson stage of the cell. These cells become inactive, but whenever we need the cells during injury or damage, we may need new cells. This stage in the cells in the Poisson stage will again divide and produce the required number of cells, thereby healing the wounds, thereby thereby correcting or repairing the damage caused by wear and tear due to the environmental influence. Organisms undergoes constant wear and tear, constant damage of their body parts, and this constant damage that develops due to interaction between the environment and the organisms is corrected and repaired by a group of cells called the Poisson cells. You got it? Now, what is equational division and what are the stages of it? Mitosis is called the equational division. Why mitosis is called equational division? It is called equational division because the chromosome number in the daughter cells remain the same as that of the parent cell. Even after the mitotic division and two daughter cells are formed, we can understand that the daughter cells have the same number of chromosomes as that of the parent cell. So in short, the parent cells and the daughter cells have the same number of chromosomes. This is why mitosis is called equational division. And what are the stages of mitosis? We can say. First, the karyokinesis, and second, cytokinesis. Karyokinesis involves 4 stages. What are they? Prophase, metaphase, anaphase, and telophase. Prophase, metaphase, anaphase, and telophase. These are the 4 stages of karyokinesis. Whereas cytokinesis is the division of the cytoplasm into 2 daughter cells. That's all. Now, explain the stages of mitosis. As you know, mitosis has 4 stages. What are the 4 stages of mitosis? We know prophase, metaphase, anaphase, and telophase. The mitosis divides generally into 2 broad steps: karyokinesis and cytokinesis. In karyokinesis is divided into 4 steps. What are they? Prophase, second metaphase, third one anaphase, and 4th stage is telophase. You have already studied and we sent the video of detailed changes that happens during these 4 stages. Once again I will repeat it. In prophase, the nuclear membrane and nucleolus gradually disappear. Nuclear membrane and nucleolus gradually disappear. The thread-like chromatin condenses gradually to form distinct chromosomes. And third step, the mitotic spindle start developing. Mitotic spindle start developing from 2 equators. So what are the 3 changes in prophase? In prophase, we can understand that the nuclear membrane and nucleolus gradually disappearing. Okay? Nuclear membrane, nucleolus, and those membrane bound organelles are also gradually disappears. Second step of this prophase, that the chromosome gradually condense to form distinct chromosome, rod-shaped chromosome. And third step, we can say that the mitotic spindle formation begins from the poles. These are the 3 steps in the prophase, 3 changes in the prophase. Now in metaphase, we can see the nuclear membrane, nucleolus, and the membrane bound organelles completely disappear. Okay, completely disappear, and the chromosome that already started becoming distinct or rod-shaped now completely compact and discrete unit of rod-shaped structures. Such discrete units or rod-shaped structures, we call them chromosomes. So chromosome completely formed in this metaphase. And the compact chromosomes are arranged at the equator of the cell, equatorial plane. Okay, that is called a metaphase plate. So the chromosomes are arranged at the equatorial plane at the metaphase plate at the center of the chromosome. That's all in this metaphase. Now in anaphase is the shortest phase in the cell cycle. And in this phase, the chromosome split at the centromere. Chromosome split at the centromere and the chromosomes start moving towards the poles. And in the telophase, you can see the moving chromosomes already reached the two poles of the cell. The chromosomes, again they decoil and become chromatin. Nuclear membrane and nucleolus start, cell organelles reforms. So in both the sides, two nuclei are formed, and the cells develop the organelles gradually, and the cells start dividing through the center. This includes the telophase. So telophase, before ending the telophase, cytokinesis also takes place. So during cytokinesis in animal cells, you have already studied the center of the cell at the equatorial region. The cells on both the sides, they develop furrows. They develop furrows and deepen the furrows, and the furrows touch, and the two daughter cells will be separated. So through the furrow formation and deepening of the furrows through the center of the equator of the cell, that indicate the cytokinesis in animal cell. Whereas in plant cell, not the furrows. Cell plate will be formed by using midlamella. With the help of midlamella, cell plates is formed at the center, and that cell plates extend towards the sides. And finally, the cell plates join the two ends of the cells, and the two daughter cells are separated. This is all about this module. Thank you very much.